So a big welcome to Stephen Goldstein and Christian Anderson, who's surprised us by just coming back into our pandemic Zoom room. You both are authors of this, I think, a definitive critical review of the origins of SARS-CoV-2, which is a pre-proof with Cell. And yeah, there's a lot of excellent material in this, and I think it's both an accounting of some of the genomic evidence, but also where the literature is at this moment in the pandemic. I've got a lot of questions. Folks who are joining, as always, feel free to type any questions in the chat box, unmute yourself, or raise your hand. Maybe I'll start out with a question relating to interpretation and maybe also language. The title of the paper has origin in it. And in the paper, you basically point to gaps, evolutionary gaps between SARS-CoV-2 and the closest related animal viruses, the widespread genomic recombination, the fact that RIT G13, which was in the Wuhan lab, there's really not a plausible pathway for that to become the pandemic virus. But I think you are also identifying two different strains of the virus. One with apparent links to the wet market at the center of all the controversy. I believe denoted lineage A and also lineage B. Or wait, am I getting them backwards?
I think that's backwards. Yeah, lineage B is associated with Wuhan on market.
So maybe just start there. I mean, and I guess part of my question about that, language of origins, like in history or the humanities, and this is maybe going to get a little theoretical. There's a philosopher, Michel Foucault, who talked about the importance of emergence rather than origins, rather than trying to pinpoint a single event, trying to think about the constellation of forces or contingent events that might give rise to a new form of life in this case. So just like sticking with the zoonotic origin idea, will we be ever able to pinpoint a single moment of transition, or do you think there's evidence here of multiple chains of transmission where it's very difficult to identify an original form that was in one particular species, or is it the traffic of multiple strains across multiple species?
That's a good question. I think it gets right to the heart of an important issue here, which is, what do the earliest events of the pandemic look like, and can we use genomic information that we have to try to uncover those? So the lineage B, as you mentioned, is associated with the Wuhan market, and then lineage A is, we have fewer sequences of lineage A from very early in the pandemic, but we know that there were these two lineages circulating contemporaneously in Wuhan by December of 2019. So an important question is the relationship of those to each other. Do they represent, as you suggested, they could independent introductions into the human population, or is there one introduction to the human population and then one of the lineages evolves from the other through the course of human transmission? And this is a key question, and Christian has been working on this directly, so I'm sure he can comment as well. But we know, for example, that in the case of SARS-1, back in 2002, 2003, that there were multiple markets that were affected. And that's probably because, as many of you probably know, in June of this year, there was a paper that came out describing live animal sales in Wuhan animal markets, including the Wuhan market, in 4 different markets in Wuhan, which I'm sure is not exhaustive of every market in Wuhan, but was a nice snapshot of what was going on in the Wuhan animal trade in the period immediately preceding the pandemic. They were in those markets studying right through November 2019. And what they found was that these markets were sharing supply chains. So it's not only is it, I think, likely that if you get infected animals in the supply chain, it would be almost inconceivable that you would only have infected animals appear at one market. So that relates to the question of whether these lineages could have essentially split before the virus entered into humans. It's not definitive, because you could have a large population of animals infected all with one lineage. But we know also from SARS-CoV-1 that there were multiple markets infected. There were multiple lineages of the virus in different animals in these markets. And so the potential existed for the kind of dynamic to exist that, Evan, that you referenced, which is the virus kind of passing back and forth between animals and humans, different lineages of the virus between animals and humans kind of bouncing back and forth. Thinking of maybe the population of the virus in the animal reservoir instead of as like a point source for introductions into humans, but as sort of like a fire throwing embers off into the human population. And some of those embers will catch in the human population and initiate new chains of transmission, some of which will die out, some of which may establish onward, continuous transmission, which is how you end up with a pandemic. Let me stop there before I keep rambling and let you kind of focus me back in, or if Christian wants to weigh in.
That was great. Yeah, Christian, if you want to jump in, if you have any thoughts.
Yeah, I mean, I think, and actually Jonathan here is the first author in that science paper that showed exactly what Stephen was just talking about, where, you have a lot of tries and most of them don't go anywhere. And this is the thing we often see, right? I mean, my group, for example, described exactly the same for the Zika epidemic here in the US, where the virus comes over many, many, many times, but most of these transmission chains, while they try to get off, most of them don't get a chance to get off. And SARS-CoV-2 is probably the same, and we certainly know that SARS-CoV-1 was that exact same. I'll say the whole distinction between, you do have two lineages, A and B, early on, and B is only found in the, or B is what we find at the Wuhan market, but it's important to understand that the Wuhan market almost certainly had a super spreading event. And would amplify up whatever lineage was causing that, presumably lineage B. So the fact that we don't see A at the Wuhan market does not mean that the lineage A wasn't at the Wuhan market, because we have very, very limited sampling from that place. And I think a lot of people have sort of misunderstood that. So I think it's important to understand, first of all, we might have both lineages. Of course, lineage A, we know from the WHO report, it's described as being associated with another market, but we don't know what that other market is. And we have tried to track it down and we can't figure it out. But if you have a situation in which you have two lineages associated with different markets across the city, and then really importantly, that you don't have any distinct intermediates between these lineages that you see in the human population, is quite strongly suggestive of two introductions that were successful. Now, I will say that I think a lot of people are pretty convinced by the data as it is today. I'm not. I think that maybe that is what occurred, and I will say that the data is somewhat suggestive of that, but there's a lot more analysis we need to do to try and understand that better. And hopefully more sequences from the very earliest cases would really be helpful here. And we unfortunately don't have any.
Jonathan,
I was just going to say that I agree. I'm not convinced yet that it's clear that there are two lineages represent independent.
Which floors can I go to?
Oh, sorry. Go ahead, Stephen. Yeah.
No, I think I would agree that if there are two independent introductions, that's clearly consistent. That's definitively for me of a zoonotic origin for the pandemic. But I agree with Christian, we can't quite get there yet. But he's, his group, he's being modest, is doing some of the work that the ongoing analysis to try to better understand those earliest events.
Yeah,
and see whether
trying
are independent introductions.
Yeah, and I would say like one thing, and Jonathan and Andrew of course know this well, but because if you look at the data set on GISAID, there are actually what's intermediate between these A and B lineages that we see in humans. And if that indeed, if we see those consistently, is strongly suggestive that the evolution of either A to B or B to A happened in the human population, which is fully consistent with a single introduction. But the problem is that we noticed issues with these particular intermediate genomes, and Jonathan has written a nice post on virological, and that's some of the stuff that we're working about, that we actually think that a lot of these, if not all of them, are due to bioinformatics errors, where they wrongly get called, because we're only talking about a single base in a 30 kb genome, right? So it's a very, very, very small error. And in fact, these kinds of errors happen all the time. But of course, because we have so few genomes from the very beginning, little errors like that can go in and make quite big differences in the analysis. So that's what we're looking at closely, because I think it's important that we get this sorted out. I will say, like Stephen did mention this, but of course, for the SARS-1, markets were not shut down and trade and farming continued unimpeded, which is not the case for SARS-CoV-2, right? SARS-CoV-2, as soon as that realization, of course, the markets were shut down, and later on in February 2020, the wildlife farming was also banned. And I can understand that ban is actually in effect in the sense that many of these animals are no longer farmed and also not sold at markets. And that sort of cuts off the transmission chain if it's actually coming in via infected animals, right? That all of a sudden, if you cut that off, you might not see additional spillovers like we did for SARS-1, which we continued to see in 2003, for example. There were several additional ones, which we have not seen for SARS-2, simply for the fact that there haven't been any opportunities.
So Mark's got his hand up. Before we go to him, one quick follow-up question, Christian. So you mentioned the single base pair, either error or mutation. Or is it a case where the samples still exist and you can go back and resequence and see if it was a sequencing error, or is that not going to be answerable?
It's answerable, but in reality, it's very difficult, right? We are trying, and we are in contact with people. We're hoping that it will be possible. We're also hoping that there are samples that exist from early cases that might not have been sequenced yet. But the reality of the situation is that we're now quite far out, right, from almost 2 years from these very early cases, but also, so stability of the samples, but also just being able to retrieve those samples, actually sequence from those samples, there being the willingness to do some of this work too from the authorities there. All of this makes things difficult. And just to compare, for example, the early situation in the United States, right? I mean, we have extremely few sequences from early cases in the United States, and the vast majority of cases went undetected even here, right? So it's not that, it's not that unusual that you have very low coverage early on in a pandemic, of course, but we are hopeful that at least some additional work can be done.
Mark, go ahead. Oh, you're muted.
There you go. I didn't want any feedback coming from you talking. Just to spell out the significance of these transitional genomes possibly being mathematical computer errors. I just want to be sure I fully understand the different interpretations, so that if these are real, then that would discredit the idea that A and B are separate lineages. Is that correct?
It would discredit. Go ahead.
I wouldn't actually say it would discredit it, because of course people can also be infected with intermediate genomes, right? If they're infected from animals, that why the whole pool is present there. But I'll say that if we do see these, bona fide intermediate happening over time in humans, yes, this suggestion that you have two unique independent introduction that managed to take off is certainly a lot less likely. And I will say that then the data becomes fully consistent with a single introduction into the human population.
Single success.
So therefore, so just to clarify,
successful.
Yes. So if it's computer error, that means that this thesis, this scenario, is still on the table. If these are actually computer errors, it's possible these are two separate lineages. Yeah, is that correct?
That is correct. Yeah, and I'll say the computer errors. I mean, my lab here developed IVA, which is one of the main tools that is used to call consensus genomes for this type of sequencing data. And we actually discovered earlier this year, so 2021, when we were working on a paper around B117, we discovered an error in the Illumina pipeline. So Illumina's own software actually led to exactly the kind of error that we believe we might be seeing here, that we discovered in 2021, and of course reached out to Illumina, and they've since got it fixed, right? But it's quite common that, because it's new ways of sequencing, a lot of people haven't sequenced before, like all these things, that things like these kinds of error crop up, and we suspect that some of the early software had indeed this particular issue, and it's down to a single tool. It's called BCF tool, and it had a bug in it, and that is then used in many different pipelines, including our own. But we had detected the issue that led to this particular error. So we think we have nailed down the potential bioinformatics error that might have led to this. But
I don't want to dwell on this, Stephen, but what is the range of error that might be plausible, possible, given these bioinformatics glitches, if I can call them that?
I mean, errors should always be expected, right? I mean, that's right. Why you can't, because again, they happen. And if we just look at the larger data set on GIS8, for example, I mean, there are errors in that data set, right? Contamination, like wrongly called genomes because of, earlier software pipelines and things like that. I mean, things get quite messy, and I think the importance is to be aware of these issues, right? And sort of be a little bit skeptical about, like, well, if I'm interpreting my data in one way or the other, am I sure that, X, Y, Z, for example? Yeah, it's very common, especially like the Zika data set, for example, was a virus that was extremely difficult to sequence. Yeah. I mean, that whole sequencing data set is filled with errors as well.
Okay, I didn't know that. Thank you.
Yeah.
Thanks much. I'll go ahead and ask another question unless someone else wants to jump in. Wim, you just unmuted yourself. Do you want to jump in?
Yes. I was interested whether maybe Christian knows how many samples would still be available in China that are unsequenced. Do you have an impression of that, or all the different animals? I mean, that would be interesting, because you might see transmissions that you do not expect, or you might find an intermediate host if you sequence more of those samples.
Yeah, I don't know. I don't know, Stephen, if you have any sense of what may or may not be available. I mean, I honestly don't know.
No, I mean, I think one of the big outstanding questions is, when the markets were shut down, were samples actually taken at that time for testing, either diagnostic or for sequencing, and that information hasn't been, made publicly available, but I have no insight into that at all.
And also other potentially susceptible animals at the same time in other parts of China, because some species are likely to be susceptible, like the martelids and so on.
Yeah, I mean, that's a big question, right? Because, like, as we now know, is that raccoon dogs and civets and other animals that we knew were certainly susceptible to SARS-1, and for raccoon dogs we know they're susceptible to SARS-2 as well, were clearly present, as Stephen mentioned, across markets, across Wuhan, but China has denied the presence of these animals, and it's plausible that by the time Wuhan CDC, for example, came to that market, they might have been removed. I don't know, but the big open question there is, were samples taken from those types of high-risk animals? Were they traced back to the holding centers, where they then traced back to the farms, and where both animals and humans tested all along these supply lines, is extremely important. And I'm not convinced that these have been tested, and certainly officially no information is available on any of that.
Yeah.
So I think to answer many of these questions, we need a small team of multi-species ethnographers on the ground in Wuhan and elsewhere. But I've actually got a question that's more kind of a history of science question and a history of very recent science. So to me, it's also a question maybe about geography and the ways that, knowledge and information gathering has been focused by, the ways that virology has been institutionalized in some cases. So in short, I'm wondering, does the detection of this in Wuhan just reflect a concentration of virological expertise, like people who had the tools and resources readily available to notice a virus that might well have come from elsewhere?
I think that, my understanding is that the Chinese public health system is set up, like since SARS-1, they set up a surveillance system specifically to detect essentially pneumonia of unknown causes, as this was originally described. So I think that it likely would have been picked up, in any Chinese city had it happened. I don't think it had to be Wuhan, but it's certainly possible that there could have been early cases in outlying areas of the province. For example, if there were people associated with the wildlife farming or the wildlife trade who got sick, I could imagine that those kinds of sporadic cases of that sort might have gone undetected early on until the virus does, make its way into the city, where it starts showing up in major hospitals and things like that. But I don't think, I think the Chinese surveillance system is equipped to detect this and this sort of event anywhere it might occur, in any city at least.
Yeah, I totally agree with that. I mean, even when I was on, which was early in the pandemic, we were sort of speculating a little bit about that. Could the detection be there? Because, well, that's why you have the expertise. I don't think so. I mean, I agree with Stephen that maybe there could have been very sporadic infections, maybe along the lines of the wildlife trade, for example. But none of these were the ones that took off. I think both the timing that we get from the genomic data and the events. I mean, clearly, as we show in our paper, and that's from the WHO report and other reports, right, is that the Wuhan market is the early epicenter here. Like all the hospitals realize that cases are associated with that market specifically. That's the first alert that goes up. We know that the early cases, when we look at those, cluster around the Wuhan market as well. And then based on some data from the WHO report that was interestingly included for completely different reasons, of different purposes, what we noticed is the excess deaths data, which also clusters. That's obviously less resolution, right? But clusters where the Wuhan market is, and again, north of the river here. So you put all these things together and saying there is a very strong early association with the markets, and specifically the Wuhan market, where you have the early cases. And that is and that very strongly suggests that, look, this is here where it took off, right? So I think it's likely that it was Wuhan, and it was definitely the Wuhan market too.
So we haven't, touched on the lab leak hypothesis yet, and in part because we've kind of talked it to death. I see Rachel has her hand up, so I won't ask that question then. Go ahead, Rachel.
Thanks, Evan. I was actually going to go in that direction. Thanks to everybody for being here, and Stephen. I guess the question that I really wanted to frame is two-part, and I agree with my colleague. I mean, Evan mentioned that this is a definitive paper, and this is a really important piece, and not least because it refutes the lab leak narratives that continue to persist in popular discourse, and not just. You say in the earliest pages of the paper, all previous human coronaviruses have zoonotic origins, and this one has several signatures of these previous events. And I think this is such a profound thing. And so I'm wondering if you can talk to us a little bit about what do you make of this sort of popular speculation narratives based upon doing this research, the experience of doing this research with your colleagues. Why does this persist? What do you make of it? And then the other question that I had was related to evidence of genetic distancing. You talked about genetic distancing, and I'm wondering, for non-virologists, what does this mean time span wise? What kind of time span are we talking about here when you talk about this degree of genetic distancing that you've found in your scholarship? What does that mean in terms of the evolution or mutation of a virus?
So I guess starting from the top, and you can jump in and redirect me if I get off course. I think the lab leak narrative, I think, persists for a variety of reasons, some of which I think are sort of understandable from a public standpoint. I think the pandemic is obviously a really extraordinary event. And so to some extent, it feels like something extraordinary must have happened, not just this sort of everyday contact between humans and wildlife that is constantly going on in the background of our lives to spark such an extraordinary event. There's obviously, at the same time, there's also a kind of news sub-ecosystem or little media ecosystem that has trafficked in sort of the particularly more extraordinary elements of this, claiming that there's a part of a bioweapons program, or alleging cover up with various scientists across the world, like Christian, for example, being involved in covering up this sort of thing. So I think there's a combination of sort of public, in good faith, kind of grasping for answers as to how something like this could have happened. And it seems like an extraordinary coincidence that there is a major coronavirus lab in the city where this all started, combined with, I think, a little bit of profiteering among some elements of the media, and a few bad actors in the sort of adjacent scientific spaces have sort of pushed to keep this very much alive. And then I think some people who are in adjacent scientific spaces who are just not kind of lacking the subject matter expertise to really filter out things that seem, relevant. We tried to address some of that, in our paper, talking about like the virus RIT G13 that was discovered by the WIV, but turns out to be, 4% nucleotide difference from SARS-CoV-2 across the span of a 30 kb genome. As Evan said, there's no path to go from RIT G13 to SARS-CoV-2. But if you're a layperson or who hears, well, there's a 96% identical virus in the lab near where this came from, it makes sense. So I think it's a complicated kind of milieu of information and motivations that have kind of kept this alive, coupled with the lack of a genuine and frustrating lack of transparency on the part of the Chinese government. I think misattributed in many cases to saying the Chinese government won't tell us what's going on in the lab. I think the Chinese government hasn't told us much about anything about the start of the pandemic, to the extent they know things. I mean, we know that, for example, people are publishing new bat viruses from outside China that are very close to SARS-CoV-2, and there are many fewer publications coming from inside China. I personally have no real doubt that Chinese groups are out doing this kind of work as well, and it seems to me that they're likely not able to publish the results of that work. The Chinese government as Christian mentioned, as I think we covered generally, misled the WHO team about whether there were live mammals sold in the markets in Wuhan preceding the pandemic. They have not been forthcoming about what kind of animal sampling or or studies of humans in that wildlife supply chains that may have been done. So I think this lack of transparency actually points to the Chinese government trying to wanting to cover up an involvement with markets. But I think the general lack of transparency breeds mistrust and lends itself to conspiratorial thinking that kind of leads people, understandably in some cases, kind of leads people down the worst or the most nefarious seeming type of scenario that is just kind of all these factors have played into each other to keep this alive.
Mark, go ahead.
Oh, you're muted.
You're muted again.
Okay, sorry about that. Can I just ask you about, I just want to get something out of the way that's bothering me about the lab leak theory, and part of this goes back to the piece that I believe Nicholas Wade wrote for the Genomic Scientist. So this issue of, the 4% difference between RATG and, I think at one point Eddie Holmes said that could be equivalent to actually 40 years of natural evolution or something like that. But I just want you to explain, if you don't mind, I'm playing devil's advocate. I don't necessarily think this, but I just want you to pin it down so that I can make the argument. So one of my students asked me why, which I've been asked by my students, So in your paper, you talk about how we've discovered other bat viruses, including quite recently, I believe, in Laos, yes? Yeah. That share more of their genome with SARS-CoV-2 than RIT G13. That's correct, yes?
Yeah.
But you have a line in your paper that collectively, the fact that we found other bat viruses that share more of their genome demonstrate beyond reasonable doubt. I think that's the phrase you use in your paper. Can you just justify that? Why is it beyond reasonable doubt? Because there are other bat viruses out there that share more of their genome, that somebody may not have manipulated this.
What is it to beyond reasonable, demonstrates beyond reasonable doubt what? I just want to make sure that I...
So I'm reading, I've taken this, collectively, these data demonstrate beyond reasonable doubt that RATG is not the progenitor of SARS, with or without laboratory manipulation.
Well, the virus can't be, RIT-G13 for a variety of reasons, I think, can't be the progenitor, but certainly if it's not even the closest virus, it can't be the progenitor, because the progenitor by definition would be the closest virus to SARS-CoV-2.
Well, not if it has been manipulated in the laboratory.
Not if it's been, right, I see what you're saying.
Yeah, but it can't. I mean, the
conspiracy theorists would say, not me. That's why I'm asking.
Right.
Yeah,
not if it's been manipulated in the lab, right?
Sorry, Chris.
Yeah, the thing is, you can't. I mean, like it's like trying to convert a spoon into a Ferrari.
Okay.
I mean, the difference between RIT-G13 and SARS-CoV-2 is huge, right? I mean, this difference of 4% doesn't sound as much, but it's 1,200 nucleotides across the entire genome. And what's really important too is that it's sort of evenly spaced across the genome, except in some recombinatoric regions or recombinated regions that we have. For example, the receptor binding domain in RIT-G13 is quite different.
Yeah, mainly just the receptor binding domain for RIT-G13.
Right. Where like the new banal viruses from Laos, for example, are almost identical to SARS-CoV-2, like really, really, really closely related. Which I will say is what people had been writing papers about, that these recombinations likely happened in bats prior to the lineage that gave rise to things like RIT-G13 and SARS-CoV-2 and the banal viruses, that the lineage leading to RIT-G13 probably lost this particular version of the receptor binding domain. And we're now seeing exactly that with these banal viruses. And one of these banal viruses is now the closest on average to SARS-CoV-2. And over the so the closest, and there, if you take recombination into consideration, there are 3 other viruses that have been detected that are also closer to SARS-CoV-2 than RIT G13, none of which are from the VIV. Those other 3 were from Yan'an, but this new one is from Laos, right? So completely different. So there's just, you know, again, there is no way in which you can turn SARS-CoV-2 into RIT-G13 into SARS-CoV-2 in a lab. That's just not how it works, right? It just cannot be done. Now, could they have had other progenitor viruses that we don't know about, and that they haven't published on, and that, you know, have told nobody about? Of course, anything is possible, right? But I think, and I totally agree with everything Stephen just said around, you know, why does this keep to persist, right? But that's why it's so important that we stick to what do we actually know, not like what could potentially be going on that we don't know about, and then we speculate, and then it's innuendo and it's conjecture, right? Which is a very dangerous path to come up on, I think, because there is actual evidence that's right in front of us. Things like the timing.
Yeah.
Probably November of 2019, right? SARS-CoV-1 happened in November of 2002. Both were associated with wet markets, where they sold things like civets and raccoon dogs. Exactly the same thing we see here. The early cases, the early deaths, the associations at the hospitals all cluster around the wet markets. There are so many things that we now know to be actual evidence that any theory you might try to pose that would explain the emergence of SARS-CoV-2 obviously have to be able to explain all of those things. And from my perspective, a natural emergence scenario explains all of this really well. A, because we have seen it before. B, we have been warned about it. And we have seen all the different components, and the virus is a unique virus, right? That is highly capable of causing things like a pandemic we can now see, right? So you have all the ingredients here that really line up and explains very well what's going on here. But any version of a lab leak would have to require a pretty substantial cover-up and lying from the Chinese scientists that have been accused of this. But also importantly, how did they get that virus? Why did they work on it covertly prior to the pandemic, right? Because prior to a pandemic, why would you not tell your colleagues about it? Like, there's all these things that are quite complicated. And then, of course, getting from the lab over to a wet market of all places, right? I mean, it could have been anywhere in Wuhan, where there's certainly other places where there are way more people congregating and you could cause a super spreading event, but it happened to be a wet market. Is that we need to take all these things into consideration as we consider what do we consider to be the most likely explanation for the emergence or the origin of SARS-CoV-2. And nothing has been proven, right? I mean, we can't prove anything yet, and very likely we never will be able to. But that's why the theories persist, right? Because it's a rare event, it's an unusual event. Pandemics don't happen every Monday, right? And we're trying to get to answers, which I think is a very natural thing.
Thank you. Eben, will you allow me a quick follow-up?
Sure.
So I'm fully on board that every epidemic and pandemic that we know of in history have started through natural causes, and disease ecology explains most of these events. But just staying with Nicholas Wade's article, the other thing that really I think is kind of one of the most important points that was made. I agree, everything you say clearly doesn't understand, you know, how you would convert a sort of lab isolated virus into a pandemic virus. But one I think the more interesting points he makes is that, you know, what we're now 20 months into this epidemic, this pandemic. I'm right in saying that nobody's produced any serological evidence, have they, that anyone in Wuhan had been exposed to the virus prior to that first case in 2019. Do you think that's odd? Because as I understand it, during SARS in 2002, 2003, there was a very intensive search, a sampling of serology, and plenty of people were found to have been exposed to it before it was detected.
Well, there was the serology I know of from 2003 was specifically, and could well be some that I'm not aware of, but there was certainly evidence that people in the wildlife trade were heavily exposed to SARS-1, occupational exposure, because of studies done by China CDC. I mean, I think it's odd to the extent that I think it's odd or unfortunate that the Chinese government hasn't been, essentially rejected the phase 2 WHO plan. The phase 2 of the WHO was supposed to include a lot of these studies, going back and doing serology on banked blood samples, tracing back through the wildlife chain, supply chains, and things like that. And so there's clearly a question about whether some of that has already been done by the Chinese government. And if so, should be
clear. We just don't know whether they've done that or not.
We don't know what, I mean, I haven't heard them say we haven't done it.
Right.
I haven't heard them say, they certainly have not released it. I have questions about whether they've done it, but those things should certainly be done. And I think it's unfortunate, and it's a major sort of pillar of the lack of transparency that we've seen from the Chinese government on this, that these kinds of studies haven't been done. Whichever direction they point, they need to be, these are critical studies that they was, in the WHO part 2, the first 4, Dr. Tedros laid out like 5 points for the phase 2 of the WHO studies. And the first 4 basically all had to do with human serology, animals, following the supply chains. And then point 5 had to do with the lab. And the Chinese government called this press conference to basically reject the entire plan out of hand. That was hugely unfortunate, because I actually thought that plan was pretty good and was in line with what the phase 1 team recommended should be done next. And it remains true that that's kind of where we are. We need to have those studies need to be done. And time is, the clock is ticking, obviously. And I think, like Christian said, it's going to be very difficult to ever, unfortunately, prove definitively what happened, unless the Chinese government has done studies that would show that and becomes willing to share the results of those studies. I think it's going to be difficult, but I do think it's, unacceptable, frankly, that these kinds of studies haven't been either done or shared.
Yeah, it's a great pity.
Yeah, and just a couple of points to that, because I totally agree with Stephen here, and I think it's important to understand that China is very clearly deflecting, anything that has to do with this virus having come from China, right? I mean, that's why they've been so focused on that. Now is the time that we need to look outside China, and we need to look at frozen food, and so we need to look at all these things. And I agree that we need to keep an open mind, and we need to look at, any possible conduit for this. And now we have found other viruses that are very similar in Laos, for example. So yes, we need to do these things, but it has to be really clear that the primary things we need to focus on is the early events that we do know about, which have to do with the Wuhan market, it has to do with the animal trade, it has to do with all these things, and investigate that closer, while again making sure that we keep an open mind to have all different theories on the table, right, or hypotheses on the table. I think it's really important. And I was hopeful that, the first WHO mission to China was not perfect by no means, but it was a start. And starts are important, because that means that you can build on them, and then you can go to the next stage, and hopefully at the end of the missions, you actually have some answers. The problem is that whole path blew up because of the politics that came in over it. Right, the blame game. It's much of what's going on now. And David, I think Nicholas Wade's article is a perfect example of this, where that article is not about trying to figure out what happened here. It's about placing blame, which for a pandemic is not very helpful, right? And certainly not when that placing of blame is completely misguided, because it includes things like myself. I mean, he wrote about how I've been covering up with Tony Fauci and things, which is, I mean, it's totally absurd.
Yeah.
Right. And anybody who knows how these systems would work was like, yeah, that doesn't make any sense. Right. I think it's a big problem for trying to understand what really happened here. And that's really unfortunate. I'm still hopeful, because I agree with Stephen that what was suggested for the phase 2 of the WHO mission were good suggestions. I'm still hopeful that we can get back on that track and hopefully do some of that. Just one final technical comment. The first cases we know about are almost certainly not the first cases, right? We know that from our modeling studies. We know it from things like Jonathan's studies showing like these dying transmission chains, things like that. Those are just the first detected cases. There are almost certainly thousands of cases that happened prior to any of those that we simply just don't know about.
Right, which kind of underlines that if they collected any blood from, October, November, and it's stored somewhere and people have antibodies, that would be great evidence.
Yeah,
absolutely. Yeah. I mean, especially in November, I would say, and of course there's these studies from Italy, and I mean, I don't believe them. I think the contaminations are much more likely to explain our observations here, right? But I mean, it is still important in the mindset of keeping an open mind here, right? That we do need to look at these more closely, and things like serology on banked samples from just around the time where this got started would be important to do. And again, was suggested in the first WHO report that we focus in on those.
Great, thanks both. Frederic, go ahead. You've had your hand up.
Thanks. You make an important distinction between viruses that are cultivated in the Wuhan Institute of Virology and RIT G13, which is only sequenced. And so there's no probability that the RIT G13 has leaked from the lab. But and so that's an important distinction, because we imagine that just because coronaviruses are in the lab, they will just jump out of the lab. But just to understand how it works from within, so if RIT G13 has been sequenced, can it be still modified through gain of function research? And so if there's gain of function research on RIT G13, it cannot lead to a leak or an infection?
So you can definitely do gain of function, or you can do genetic engineering based on the sequence and make the virus essentially de novo from the sequence. That is something that can be done. There's no evidence, a couple points. So with the WIV, they did most of their genetic engineering with a virus called WIV1, and that's pretty much all the evidence we have is that primarily their focus was on using this virus as the foundation, as the backbone for their genetic engineering work. RIT G13 also, people have tested the RIT G13 spike to see how well it'll mediate infection of human cells, and it does very poorly. It does okay on mice and rats, so it's different than SARS-CoV-2, but it's a very poor candidate for a virus to actually work with in a human cell culture system. So I think, there's no evidence that they were doing any kind of like gain of function work with RIT G13. There's no evidence that that virus has been engineered or cultured anywhere in the world that I know of, just the spike protein. But it appears that it would grow pretty poorly on primate cells, pretty much all of which are the standard cells that are used to grow these viruses in the lab. The main cell line that's used are viro cells. And then often different human cell lines will be used if we're interested in how these viruses are doing in infecting humans. And RIT G13 seems particularly poorly adapted for use in those types of systems. So the level of concern with respect to specifically RIT G13, I think really should be quite low for a variety of those reasons.
Yeah, I think that the only sort of viable version of this, right, is that it's not RIT G13. It's something we don't know about.
Yeah.
I would say gain of function on any of the viruses that we do know about could not have led to SARS-CoV-2. I mean, it just doesn't make any sense. What has to be clear to is that RIT G13 was not cultured, and according to Viv, they have 3 cultured viruses, right? Is that culturing viruses is incredibly difficult because bat samples typically have very little virus in them. They're degraded, like all these things, right? It just makes it really, really difficult. You can create whole viruses that even have no scars based on just a sequence. That can be done. But here's the thing, that if we look at the work they have done at the Viv, for example, they haven't done that before. They always use the same cloning systems. They use Viv1 and Viv16s are sort of like their to-go, and Viv1 is specifically the bacular system. The system that they use for their cloning. Then they pop spikes into that one, right?
Yeah.
And if you look at these 48 reports of annual reports from their NIH-funded work that came out, as they have been saying that indeed it's Viv1 over and over and over again. Right, because it works. They have it established in the lab, it works. And it's a back system. They use cloning enzymes, restriction enzymes, and things like that. And SARS-CoV-2 have none of those features that you know that they have been working on and using in the past. Does that mean that they couldn't have created something completely new? Of course they could. Anything is possible. But again, there is no evidence to suggest that they did, right? And I think that's, again, why it's really important is that we're not talking about like trying to make conclusions based on hypotheticals, of which their universe is endless, because literally anything is possible, is problematic, right? And again, we have to remember, yes, but then what about the wet markets? What about the association? What about the data we actually know to be true? How does that fit into a theory about them having performed gain of function that would have led to SARS-CoV-2, for example? Again, not impossible, right? But we have no evidence for it, and it seems unlikely based on alternative theories about the emergence.
Thank you. So Francisco has a sort of more epistemological, philosophical question in the chat. Francisco, do you want to articulate that out loud?
Thank you, and probably very briefly. When I read the last paragraph of the paper, I assumed that for you, Christian, Stephen, and your colleagues, a Popperian attitude towards finding the origins of SARS-CoV-2 would be the preferred one. But during the presentation, I somehow sensed that you are in contrast, entertaining the possibility that perhaps inductively we could find what those origins could have been. I don't know if you have any comments regarding that.
I mean, I think because how do we ever know anything to be true is a good question, right? If you have proof, well, then you can probably believe it to be true, right? But what constitutes a proof is pretty difficult. I can tell you that if there is any evidence beyond any reasonable doubt that the Viv was working on a virus that is SARS-CoV-2 prior to the pandemic, you've proven the lab leak to me, right? Like this is no beyond any reasonable doubt where you can say, look, that just doesn't make any sense, right? Then we know they've been lying about it, and now the virus is placed at the lab. So there you go. That's as close to a proof as you can possibly come. On the other hand, you say like, well, if we see another spillover of SARS-CoV-2 outside Wuhan, for example, of what's called a basal virus, one that's slightly ancestral to the one that actually caused the current pandemic, is as close to proof that you can come to natural emergence. But the reason why in our, the last, paragraph of the Cell paper that we warn that we have to be careful that we don't get up with a hypothesis which is unfalsifiable, because the problem is that if I find a really closely related virus in an animal market outside Wuhan, the goalpost might very quickly shift to saying like, yeah, that's the virus they had in the lab, right? And that's why we write that and saying like, we have to be really careful that the hypotheses that we build are actually testable and falsifiable. And it's unclear to me that many versions of the lab leak hypotheses that are being circulated now are in fact unfalsifiable, i.e. not scientific. And I think that's what we're trying to allude to in towards the end of the paper.
Thank you very much, Chris.
Thank you. So I opened with a comment about language and origins versus emergence, and maybe I'll make a similar comment about language and kind of trying to wrap things up. I think the word natural emergence, Christian, you just used it, and Mark, you also used it as you were asking a question, presumes like separate domains of existence. And the wet market hypothesis is a natural, industrial, cultural hypothesis. It's a particular assemblage of forces that have been brought together at a particular historical moment. So yeah, and maybe that's part of what our job is to do as the theorists of the social, the theorists of the technical and political and economic, is to remind how nature is always, at least these days, situated in these assemblages. So I think without further ado, Frederic, you had your hand up. Did you want to ask one final question? All good. Okay. Thank you so much, Stephen and Christian. Really good to have you back in the room. Yeah. I'm also pleased to report that Jonathan has offered to discuss his work in a future Zoom room. So stay tuned for that. And next week we have Charnay Lavery, who's a scholar in the Department of English, to continue kind of the interdisciplinary dialogue about these issues. So thanks again. It's really great to have, we've been at this for a year and a half now, and it's great to see so many participants and panelists who are back. We've been in this for the long haul, so it's great to have this community. So thank you all.
Thanks, everyone.
Thanks, everyone.
Bye.
Thanks, Stephen. Thanks, Christian.
Thank you. Bye.